So the second part of meiosis is called meiosis II. Now, in between meiosis I and meiosis II, there is no interphase this time, okay? Or it's a very short period of time, just there's no more DNA replication. And remember, meiosis II is going to be similar to all the stages in mitosis. You'll see as I go. So, the first one is going to be prophase 2. In prophase 2, it is identical to prophase in mitosis. So you see the spindle fibers are starting to appear, the centrioles are on the ends of the spindle fibers, and the chromosomes are starting to attach to those spindle fibers. The next stage is going to be metaphase 2. Again, the same as metaphase in mitosis. Here you see the chromosomes lining up in the middle of the cell over top of each other. And remember, these are pairs of chromosomes. Okay. Then we move on to anaphase 2, same as mitosis again, where the sister chromatids will separate. So you see here in this drawing that the chromosomes have separated from here and from here. Okay, so this is going on simultaneously in both cells at the same time. Then the next stage is going to be telophase 2, same as telophase in mitosis. The nuclei will form around those sister chromatids. Cytokinesis will occur where it pinches together and creates two separate cells. And remember, when we are talking about meiosis, that there are 4 haploid daughter cells that are being produced. Whether you have sperm or egg, you're going to have 4, okay? Here is a graphic of what telophase 2 and cytokinesis would look like. So here you have your telophase 2 going on in both of the cells that have been created. And then down here, you see where the 4 new cells have come off of those 2 original cells. Now, if you recall, when we mentioned crossing over earlier, that the other problem that occurs in meiosis is something called non-disjunction. And that is when the homologous chromosomes, or the sister chromatids, don't separate like they are supposed to. Okay? So then that gives one of those cells an abnormal number of DNA. So it can be damaging to the cell. So it can occur in one of 2 ways. Either both sister chromatids will go to one side of the cell or the other, or when they separate, you'll have one that will go one way, and the other one, instead of going the other way, kind of just disintegrates. So you have monosomy and trisomy. So monosomy would be an egg that only has 1 chromosome instead of 2, like it's supposed to. And trisomy is when on one of the pairs you would have 3 chromosomes. So you would have an extra chromosome. Okay? Here are some common disorders that are associated to non-disjunction. You have Down syndrome, you have Turner syndrome, you have Klinefelter syndrome, and you have Edwards syndrome. And if you notice, Turner syndrome that's missing the other sex gene. And in Klinefelter's, that person has two female sex genes and one male. Okay? And again, we see this idea of the karyotype that we saw briefly before. And the karyotype is the two pairs, the chromosomes matched up so that they are aligned according to size. And in this particular karyotype, you can see that there are three chromosomes, an extra chromosome, on that 21st pair. So that particular disorder is trisomy 21. So that would be someone with Down syndrome.